ladies and gentlemen, let's dive right into Chapter 3. There are 5 sections. This video is going to cover sections 1 and 2. Organic molecules. Organic molecules are molecules that have carbon and hydrogen atoms. That's really the only thing that they require. There are 4 main classes of organic molecules, also referred to as biomolecules. They are carbohydrates, lipids, proteins, and nucleic acids. These 4 categories of molecules have a very wide range of functions, and we're going to go into what those functions are throughout this chapter. Before we dive into the 4 types, we need to understand what macromolecules are, these large biomolecules. Each one of them has functional groups. There are hydroxyl, carbonyl, carboxyl, carboxyl acid. These are just different parts of molecules, and they each have different structures and therefore different functions. We're not going to spend a whole lot of time on this, but I need you to know that these different functional groups, these different structures, are what cause these macromolecules to have different properties and different functions. Here are some more functional groups: aminos, sulfohydryls, and phosphates. So all living things have carbon in them. It is a defining characteristic. Every living thing has carbon and hydrogen atoms in its molecules. The carbon atom is a very unique atom. It has 6 electrons, and it wants to have a full outer shell, like all atoms do. And to get that full outer shell, carbon will form 4 covalent bonds. And it bonds very readily with other carbon atoms, with nitrogen atoms, hydrogen, oxygen, phosphorus, and sulfur atoms. When carbon bonds with itself, it's very stable. All right, so it likes to form carbon-carbon bonds. Those bonds are covalent, and those electrons are shared equally among those 2 carbon atoms. Long chains of carbon atoms can be formed where the backbone or the center of the molecule are carbon atoms, and there are hydrogen atoms on the outside. This allows each carbon atom to form 4 covalent bonds with a combination of other carbons and hydrogen atoms. Carbon can also form double covalent bonds and triple covalent bonds, meaning that it shares more than 1 pair of electrons with another atom. So it can share up to 6 electrons with another atom. Ring structures are also possible with carbon-based atoms, which is important moving on. So just make a little note of that. These hydrocarbons, these long chains of carbon and hydrogen atoms bonded together, can form branches, making these carbon chains very, very complex, as you'll see in later slides. The carbon skeleton and functional groups. The carbon chain of an organic molecule is called its skeleton or its backbone. I generally use the term backbone a little bit more than skeleton, but they are interchangeable. Functional groups are clusters of specific atoms bonded to the carbon skeleton with characteristic structures and functions. So those functional groups determine the chemical reactivity and the polarity of that organic molecule. For example, the replacement of an H by an OH and the second carbon of a hydrocarbon chain turns ethane into ethanol, and from hydrophobic, meaning water-fearing, into hydrophilic, meaning water-loving. So just one small change in that molecule completely changes its function. Isomers are organic molecules that have the same atoms, they have the same molecular formulas, but they are arranged differently. Okay, so here we have glyceraldehyde, and it has all of the same atoms and the same ratios as this molecule over here. But you'll notice that the double bond, the 2 lines right here, that double covalent bond in the glyceraldehyde is in a different location in this molecule. So they have the same elements in the same amounts, they're just arranged a little differently. So those are called isomers. Biomolecules of the cell. carbohydrates, lipids, proteins, and nucleic acids are all called biomolecules, also referred to as macromolecules. They usually consist of many repeating units, and each of those units is called a monomer, mono meaning one. These monomers join together to make a polymer, poly meaning many. So a polymer is made up of a lot of individual monomers that have been bonded together. For example, amino acids, which are the monomers of proteins, are joined together to form a protein, which is a polymer. So it's an amino acid bonded to an amino acid, bonded to an amino acid, bonded to an amino acid, that eventually makes up a protein. Lipids are not technically polymers because they contain two different types of subunits. So lipids, while they're made up of lots of smaller molecules that are bonded together, it's not the same one over and over. So it's not technically a polymer. Here are the four categories of biomolecules: carbohydrates, lipids, proteins, and nucleic acids. Here we have a table showing the monomers, or the single units that make them up, and the name of their polymer. Notice lipids do not form polymers. Synthesis and degradation. A dehydration reaction is a chemical reaction in which subunits, so those monomers, are joined together by the formation of a covalent bond, meaning they're sharing those electrons, and water is produced during the reaction. So the removal of that water molecule causes the two monomers to bond together to make a polymer. So if we take a bunch of glucose molecules, which are our monomer, and we bond them together in this way, we can form a starch molecule, which is a polymer. Hydrolysis is the exact opposite of dehydration reactions. It's when a chemical reaction in which a water molecule is added to break the covalent bond. So hydro means water, lysis means breaking. So hydrolysis is literally breaking bonds, breaking large molecules down into smaller ones using a water molecule. So the digestion of starch into glucose monomers would be a hydrolysis reaction. So whenever you eat French fries at lunch, your body is going to go through this hydrolysis reaction and break that large starch molecule down into individual glucose molecules that your body can then use to power its body processes. So here we have a monomer and another monomer, but we're just worried about this one, and it has a hydroxide right here, an OH. And another monomer is added with just a hydrogen here. We're going to stick those two together, the hydroxide and the hydrogen, and form a water molecule. And whenever we take out that water molecule, it causes this monomer right here to be bonded with this one. And that is how polymers are formed. Here, we're going to use a water molecule to break the bond right here. So we insert it in here, and we break our molecule. Enzymes. Special molecules called enzymes are required for cells to carry out dehydration synthesis and hydrolysis reactions. Enzymes are molecules that speed up a chemical reaction. Without these enzymes, these chemical reactions could not occur. They speed it up. They make sure that all of the pieces that are needed are brought together in the correct way. They're kind of like a producer on a movie set. The producer is in charge of making sure that everybody is there that needs to be there, and they're all doing their jobs. Enzymes are like that. They bring the people, they bring the molecules in that need to be there, and they stick them together in a way that a chemical reaction can occur. Now, enzymes are special because they are not consumed in the reaction. They're not changed in any way. They start out the same way at the beginning, and they end in that same way. So they are called catalysts because they speed up the reaction. You know what, guys? I think we're going to stop here because I don't think we'll have time to get through the entire carbohydrate section. So I'm going to pause the video and start a new one.